I propose that I'll start with one question for each of you, and then that we rapidly open up to the audience. Maybe Chris, I start with a question for you. So one question is like the step from rodent to human, what your ideas are particularly about this. You said in 6 months this will have already arrived in the clinic. I assume the vagal nerve looks different. There's a lot more on the surgical side to solve. So this is one part of the question. The other question is the vagal nerve not being only involved in interoceptive coding and visceral programming, but of course it's a fantastic nerve also for almost the entire field of internal medicine. Cardiac, heartbeat control, respiration, blood pressure, for example. So maybe a quick response for the first part and then...
I think on the application of the vagal nerve, I completely agree. I mean, one of the projects we've got is looking at cytokines and looking at inflammation and looking at early detection of cancer. And I think, you know, this is where I think some of the work that Dr. Waleed has been doing with me at Imperial College, where we've been looking at the relationship between, say, early detection and genetics and some of these metabolic pathways. So that's a very important field. When it comes to the implantation, the good news is that these devices have already been implanted. So the surgical procedures are already in place. And when you look at how crude a sort of bypass operation is, something like this is pretty straightforward. Sometimes a surgeon will make the mistake and accidentally cut the vagus nerve at the actual afferent point. And that is evidence that actually the vagal nerve does suppress appetite, but obviously it has a lot of other side effects. So in terms of surgical procedure, I think we're just sitting on the back of other people that have actually demonstrated that they can do it.
But to bring your new system, I mean, this could mean more electrodes, becoming more, I think you mentioned cuff electrodes, I think, so this is a procedure that's already known, but how do you get to more electrodes and denser electrode arrays?
Well, see, the important thing here is that, you know, we can get electrodes down to the fascicle level now. You know, there are a number of fascicles in the axon, so we can go down to sort of you showed some electrodes earlier, we can get down to sort of 0.2 millimeters of dimension, which is the size of the rat vagus. The human vagus is 2 millimeters. So there's not a huge demand on the actual amount of the diameter of the electrodes itself.
Okay. One question for Stéphanie. So you showed these amazing data characterizing the spinal cord as a tube that is bending tremendously under movement, under body posture changes. But in a sense, I would assume for an electrode array to be fixed, it may be, despite this dynamic pattern, rather straightforward compared, if you particularly look in the human brain, of a massively gyrified cortex, where we have 1.5 kilograms of mass of brain matter, which also goes deeply into cell size. So are soft electrodes, the type you presented, also particularly well suited to go into cortical applications?
Yes. So really when we developed this technology, the idea was to have a technology that would be as generic as possible. So we picked, as I mentioned in the talk, the spinal cord location because it was mechanically the most challenging one. But of course, we're very interesting also in interfacing the surface of the brain, and the convolution of the surface of the brain is also something where having electrodes that are extremely soft and extremely compliant so that it can form and have access to the inside of the gyri is something we want to explore. We use the technology already at the surface of the brain in order to monitor motor intention, for example. And so this is one first step for using this technology on the brain.
So this would be an animal model, or if you look at a rodent brain or even some macaque brains, they're quite smooth still on the cortex, whereas the human brain is massively gyrified, right? And much of the growth of the neurons is because of the gyrification. And very often in the depth of the sulci you find blood vessels. So how did you deal in the previous work already, even in animals, with blood vessels and electrode placement?
So up to now we haven't folded the array into the gyri. And so this would be something we have to explore now, and this is where, again, we'll have to work very closely with the surgeon, because they're the one who are actually telling us, teaching us how the implant should look like so that they can manipulate it. So this is now a new open field for us to do this.
Okay. I have about 1,000 questions for Karlheinz Meier. I start with one. And since we have in these other talks been applied towards disease, and I'm sure you have a lot of ideas also how neuromorphic computing can be applied to understanding disease processes, I'd like to hear cases of epilepsy, for example, where you talked about non-synchronized states where hypersynchronization is a matter, and it's tightly linked to the hippocampus. So what are your ideas about disease processes and how neuromorphic computing can teach us?
Well, that's probably the most difficult question. To me, that's really far away, because you need models for diseases in order to implement them. It's also that, I mean, epilepsy, for example, and other diseases and the states of the brain circuits can, I guess, easily be simulated on conventional computers. At the moment, I cannot really see that new computing architectures are necessary to do these kind of studies. There are two areas where I see the new computing architectures are the only way to go. And this is the bridging of timescales, as I said. I mean, if you really want to link very, very slow developmental processes and the millisecond activity of individual neurons, then I think conventional computers have no chance. It's the only way to address this issue. The other thing is robotics, of course. I mean, if you want to build robots, which obviously have to be mobile to some extent, the systems need to be low power and compact. And again, that is something conventional computers cannot do. The disease aspect, I would expect also in the Human Brain Project, will be mostly done on conventional, in quotation marks, computers or even supercomputers. I don't think that is the prime application of neuromorphic computing, but I may be wrong, of course.
Concerning functions, what would be your preferred function in the nervous system that you look at independent of disease using neuromorphic computing? I think you mentioned 200,000 neurons is possible. So could one look at pattern generators in the spinal cord? Could one look at the hypothalamus controlling some of the vagal nerve functions?
I mean, the numbers I gave, both for the Spinnaker system, for our system, and for the other neuromorphic systems, those are numbers for one unit. I mean, typically the good thing about this way of computing is that it is scalable. So if you want larger systems, you just build more. And I forgot to say in my talk that the Manchester group is going to build a system with 1 million ARM cores, and our systems will have 1 billion plastic synapses, so that they will just scaled up in the first phase of the Human Brain Project, and these systems will be available later this year. They are actually at this moment under construction. The functions I would like to see implemented are the functions that I find most interesting in the brain, and that is, for example, things like temporal sequence learning, the ability to make predictions based on the data that you receive by the learning process, and also to discover causal relations in data. These, to me, are the two things which I find most exciting in neural systems, which I would really like to implement. And this is not just a crazy idea. I mean, I tried to point out in my talk that there are theories now available that you can take and you can implement and you can test. For example, for temporal sequence learning, there is a model called hierarchical temporal memory developed by Jeff Hawkins in the US. He has his company, Numenta, where he does exactly that. And the interesting thing is that Jeff Hawkins requires the things that we are implementing in our system, like, for example, structured neurons. You have to go away from point neurons. You have to give structure to neurons. You need aspects of dendritic branching, dendritic computation, dendritic spikes, backpropagating action potential. So this is what we are trying to implement. The other one is the growth of connections. That's another important thing. If you want to do learning, you have to be able to switch on connections during the learning process. This sounds very difficult and almost impossible, but we are working on it, and again, it's something that will be coming up in our Human Brain Project systems in the next years.
If anyone has a question, just have to stand up and go next to the microphone. Sir, you go first.
Michel Lecruy from Medtronic. It's a question to the whole panel, actually. When you look at the simulation of the brain and the tremendous neurocomputing and neuromorphic computing, I'm asking myself, what do we do about the biological complexity of this system that is electrobiochemical, actually? You know, gene expression, protein regulation, concentration of neurotransmitters in the fluid surrounding the neurons. All those factors must have an influence on the behavior of the neurons. How do you envision to capture that in future simulations of the brain?
I can try to answer. It's a very good and very difficult question. It's clear that the models we have at the moment are extremely simple, and we are not capturing all the aspects that you have just described. Now, the question is always which of those aspects are necessary to achieve a certain degree of computation. I mean, I give you an extreme example. People in the past have done classical neural networks, like the perceptron network, for example, which was extremely simple. It was not at all biological, and still it captured some very, very interesting features of neural systems, like that there are layered structures, that there are synaptic weights, that there are learning mechanisms, and people are using it today. For example, Google is using, with deep learning is nothing like classical neural networks trained with very powerful computers. So my claim is that as soon as you get some aspects of the brain, you can always do interesting information processing. We will never be complete, but the question is which part of biology are essential and which can be left out. So what is the strategy of the Human Brain Project there? I showed the computing strategy, and we have these supercomputer simulations in which we can put very, very detailed cell models, for example, even including things like individual ion channels, maybe even going down to gene expression. So that can be put on a supercomputer, but you cannot, with that kind of simulations, I think I've shown that clearly, you cannot simulate dynamical processes that run over days or years. So what we will do is to put very detailed simulations on supercomputers and then in a systematic fashion reduce the complexity, make the models simpler and simpler, and take the simple models and put them on the special computing architecture and then see what we can capture. So there is a systematic process of reducing complexity, and I think this is what we have to do. In terms of neuromodulators, for example, slow processes, we have plans to implement that very soon on our neuromorphic systems, actually. That will happen very soon.
I think, Chris, you...
Yeah, I was just going to say that I've worked with a chap called Carver Mead about 20 years ago at Caltech when he first coined the word neuromorphic engineering. And for me, it was just an alternative for analog computation, because instead of crushing the physics of semiconductor devices into ones and zeros, what we do is we use the physics to do the computation. You use the exponentials, the logarithms. It's the old analog computation, and therefore you've got more computational efficiency in a single device. If you start combining that now with Nernstian physics and the relationship between pH and hydrogen ions and pyrophosphate right down at the sort of nanostructure level, then you can look at metabolic pathways, you can look at integrating these systems and making things a lot more intelligent locally. And I think that's the way things are going. I think it's that whole integration. And it will only happen, actually, we're only learning this now, when you've got a biochemist rubbing shoulders with an electrical engineer in a laboratory. That's the only way these things are going to happen.
Thank you. Sir.
Yes. Salah Basalama, HBKU Qatar. I'd like to ask a question, Dr. Chris Toumazou, about, you mentioned that there is an existing cuff, electrode cuff that is wrapping the vagal nerve that is used to prevent seizures. I'd like to know, and you mentioned that there are side effects. Could you tell us more about the side effects of the cuff that you are using or you want to use on the vagal nerve for the obesity problem?
Absolutely. Well, the side effects are sickness, drowsiness, obviously, headaches. And the reason being that, see, if you connect the cuff to this branch of the vagus nerve that goes from the brain, we call it the efferent part of the vagus nerve, and you stimulate the brain that way, then it's not selective. You don't know where you're stimulating. You don't actually know which fiber the electrical stimulus is going down. And that's why you've got all these awful side effects. There's no selectivity at all. I'd like to make it selective, and that's why I want to be able to measure the calcium ionic changes as a result of an epileptic seizure, and to only stimulate based upon those intelligent chemical reactions. However, what we're doing for obesity is we've moved away from the general microprocessor bus, if you like, from the general fiber, and we've actually gone right down to the afferent part, the bit that actually connects to the gut itself. So now you're wrapping the cuff around the part of the fiber that actually is connected to the gut. So the signals that are coming from the gut, the hormonic signals, are then directional. They're selective because that's all that's going through the cuff. Once you know those signal patterns, then you can stimulate and it will go back to that part of the brain. So you've effectively made it more selective by homing in on the part of the metabolism that's very, very relevant to that particular disease.
I had maybe just a quick follow-up. I had a similar question. How do you become more specific, distinguishing between outgoing and incoming signals? So these electrodes, cuff electrodes, or I think you also presented other electrode systems, they are, of course, targeted, but then the decoding depends on what kind of signals you have, right, on the fibers you are close to. So what's the percentage of electrodes that you can really use? I assume since you can't target it, it must be rather low.
Yeah, no, we can integrate something like 10, 20,000 ISFETs. A microchip. On this here, I've got 4 billion ISFETs on this chip. So these are just sort of integrated transistors. So the amount of averaging that you can do with a number of sensors is enormous with microchip technology. But it's actually making those sensors selective to the ionic changes that you want to detect due to hormone release. If you want to detect the ghrelin hormone, for example, which is the hunger hormone, then you can measure various calcium and sodium ionic reaction patterns that occur. And what's really nice, and the big difference is, by the way, when you measure chemistry, you don't have the problem of muscle noise. When you're trying to measure electrical signals, that you've got muscle getting in the way, and muscle movement actually causes a lot of signal to noise. A lot of noise, effectively. So by measuring chemical reactions, you don't have that issue with noise that you get with electrical activity. So it's much more selective.
Maybe a similar question for Stéphanie, how to make your stimulation device selective. So measuring and of course getting locomotion, robotic input is very important. How does this drive the stimulating input, and what do you see as a number of maximum electrode, let's say in the human case, that you would like to have in the ideal case in a dorsal column, a spinal cord implant?
Well, I think this is still an open question at the moment. We are just discovering by distributing electrodes, just a few of them. At the moment, we only have up to 8. We are already able to actually refine and stimulate and control both legs and also height of the different movement. And so I don't think it's a matter also of the number of electrodes. I think it's what is very important is where precisely we can locate the electrodes. And this is at the moment very open. We haven't yet finalized such designs, but because also the higher the density of the electrode, the more challenging, of course, the control loop will have to be. So I can't give you a straight answer on this.
One last question, sir.
This is Bragi from Neurotechnology. As we all know, a computer architecture is a physical implementation scheme of two mathematical constructions. the computational model that defines how to process information, and the information theory that defines how to represent or encode the information. So my question is it ever possible to move away from the conventional von Neumann architecture without changing the underlying Turing machine and Shannon information theory? Thank you.
Well, there are many aspects here. You talked about the Turing machine, you talked about the von Neumann architecture, and you talked about Shannon information. I mean, these are very different aspects of information theory. The definition of Shannon information is that the measure of information is the negative logarithm of the probability for a certain information to occur. And that, of course, is present in neural systems as it is in any other system. So you can use Shannon information to quantify the amount of information in a neural system. The von Neumann architecture is an implementation of a Turing machine. It's an architectural implementation. And the essential feature of the von Neumann architecture is that there is a physical separation of memory and computing. And that is something we are giving up at this very moment. In the neuromorphic architecture, there is no unit for memory and for computation, but the two functions are interleaved. I mean, that is something we are doing today. The Turing machine is an interesting question because it's a mathematical model of processing information. And the question whether the brain or any physical model of the brain is a Turing machine is not so easy to answer. I mean, the implementation is not a Turing machine, but it may well be that it can be emulated by a Turing machine. But that question is not answered, and many scientists are thinking about this, and with the computing system that we build, I hope we can contribute to that question. That is a really very fundamental one. One final remark, you said we always go through mathematical abstraction. This is true for the time being. So we look for biology, we analyze biology, then we write down differential equations which represent a mathematical abstraction, for example, of a cell response. And then we go and simulate it either on computers or we build a physical model. On the longer term, I hope we can go away from this intermediate step of the mathematical abstraction and go directly from the biological information to the physical copy. I mean, that would be true neuromorphic computing.
Thank you to all the speakers for their contributions. The session ends here. I think they all deserve a big applause.